Last time we began a discussion about biological macromolecules, and these included 4 different categories of substances. You had proteins, you had carbohydrates, you had lipids, and, last but not least, you had nucleic acids, which is going to be the one that I start out with today. There are 2 main types of nucleic acids that you find inside of cells. One of them you're probably very familiar with because it makes it into the news all the time. People talk about it constantly, and that is DNA. So first of all, what does DNA stand for? It stands for deoxyribonucleic acid. So the Na is nucleic acid. That's a pretty big hint to remember which category of macromolecule DNA fits into. It is a double-stranded molecule. So here you can see my rotating model of a DNA molecule, and it kind of looks a little bit like a spiral staircase, doesn't it? You have these 2 strings going around the outside that circle around each other in a counterclockwise spin. Each one of those strings is a single strand of DNA, and DNA is a double-stranded molecule, so the 2 individual strands are bound together. How are they bound together? Through hydrogen bonds. Our old friend hydrogen bonds from way back in the water video. They are what actually hold the 2 halves of your DNA molecule together. Now, the purpose of DNA is information storage. That's what it's really, really good at. It's good at storing information and copying itself. But we're going to have a whole section where we talk about how DNA copies itself and how it stores information and how that information is used to make proteins and how those proteins make other types of molecules. So for right now, just for introductory purposes, this is a DNA molecule. Its job is information storage. Then we have this other kind of nucleic acid, which is called ribonucleic acid. So RNA, again, Na stands for nucleic acid, and that is a single-stranded molecule. So you don't get the 2 halves twisting around each other, you just get 1 string of RNA. And RNA has a lot more diversity in its function. DNA only has the one job, storing information. But RNA is involved in transferring that information from point A to point B. It's involved in information processing, in translating that information, in putting together proteins. RNA has lots and lots and lots of jobs inside of the cell, and you'll see, I think, 4 or 5 different versions of RNA later on in the class, but again, introduction. So, RNA has many jobs, and it's a single-stranded molecule. Now, you may remember from the previous video that the monomers of nucleic acids are the nucleotides, and there are 5 different nucleotides. And they have the following names: thymine, adenine, cytosine, guanine, and uracil. Now, for the nucleic acid monomers, for the nucleotides, we typically just abbreviate them to 1 letter because we have to work with them a lot. So just putting down 1 little letter is fine for any kind of practical problems. So you get T, A, C, G, and U. Each kind of nucleic acid only gets 4 of these monomers. So T, A, G, C, these are the DNA monomers. These are the DNA nucleotides. A, C, G, and U, these are the RNA nucleotides. You only find U in RNA, and you only find T in DNA. That little detail is not so important in this first unit, but it will become important in a later unit, so I figured I'd let you know about it right now up front. Now, I said that DNA is involved in information storage, and you've probably heard this before, like DNA is the blueprint for making an organism. DNA is the blueprint for you. And we all have this kind of sense as like your DNA has something to do with whether or not you're lactose intolerant or lactose tolerant, or whether you have a big nose or you have, blue eyes or whatever it is. But what is the DNA actually doing? What is it storing information for? And what it's storing information for is how to build proteins. Proteins are the molecules that do really all of the work inside of your cells. DNA is the poster child. It's probably the most famous molecule in all of biochemistry, but proteins are where all the real action is. But you can't make the proteins unless you know how to put them together, and the DNA is the instructions for building those proteins. And if any of the DNA instructions are even slightly off, your proteins won't work in the same way that they otherwise would. So you could have slight changes to your DNA code, which then cause the protein, which is responsible for making pigments in your eyes, to no longer be functional, and as a result, you make no pigment in your eyes, and it turns out, when you make no pigment, you end up with blue eyes. So a little change to your DNA changes the protein, and then that changes your overall development, so it changes your traits. And I don't think anyone will be surprised to find out that DNA is the genetic material of inheritance, so it is the reason why sons look like their fathers, daughters look like their mothers, that you might have characteristics that you inherited from some from this parent and some from that parent, or maybe you're the spitting image of your great-grandfather or your great-grandmother. That all comes down to the inheritance of DNA. Now, in the previous video, I already showed you what a nucleotide looks like, but just as a reminder, you have a phosphate group and a sugar. So here's our phosphate, and then this blue pentagon is our sugar. And if you count up the number of carbons, you have 5 carbons in this sugar. So both DNA and RNA have a 5-carbon sugar, but it is a different 5-carbon sugar. RNA uses this sugar right here, which is called ribose, and DNA uses this sugar right here, which is referred to as deoxyribose. And if you had to take a guess, you would probably get it right as to what the difference between these two sugars are. Deoxyribose has 1 fewer oxygen atoms in its structure than ribose does, and here's the missing oxygen right here. Now, does that really make a difference? Not terribly much of one, but it is one of the main differences between DNA and RNA that you're going to want to remember. So we have our phosphate group, we have our sugar, and then last we have the nitrogenous base. And there are 5 different nitrogenous bases, 1 for each kind of nucleotide that there is. Up here we have what are called the pyrimidines, and they have 1 ring in their structure: cytosine with a C, thymine with a T, and uracil with a U. Remember, thymine only shows up in DNA, and uracil only shows up in RNA. And then we have the purines down here, adenine and guanine. They have 2 rings in their structure, so they're just a little bit bigger. In order to keep the purines and the pyrimidines straight in my head, I remember the mnemonic pure silver, because the chemical symbol for silver is Ag, so adenine and guanine are pure silver, they are your purines. And the pyrimidines, well, they're the other ones. I've also heard other biologists say that they use the phrase pure as gold to remember, so as gold, Ag, and the purine is in there, but I find that confusing because the chemical symbol for gold is Au, which makes me think that adenine and uracil is it, and it's just the way my brain happens to work, which is admittedly probably pretty strange, so you use whichever one you think feels more natural. So, we have different nucleotides, each one will have a phosphate group, a sugar, and then one of these 5 bases, and then we use a dehydration reaction. If you forget what that is, watch the previous video. We use a dehydration reaction to link them together, and the way that we link them together is that the phosphate group of one nucleotide is going to attach to the sugar of the one directly above it. So you get this repeating pattern of sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, going all the way down the line. Now, the nitrogenous bases are not involved in that chain, so they just kind of stick out to one side. When biologists first saw this, they said, hey, we need a catchy name for this structure, and they saw this chain of sugar and phosphates going down the line with all these little nitrogenous bases sticking out, and of course they were in a biology lab, so there was a skeleton hanging up. This is my guess anyway. And they said, it looks like the backbone. It looks like the vertebrae, the little processes that come off the vertebrae there. So we'll call it the sugar-phosphate backbone. Now for RNA, that's all you have, this sugar-phosphate backbone with the nitrogenous bases sticking out over the side. But DNA is a double-stranded molecule, so we need to know how the 2 strands, the 2 sugar-phosphate backbones, link up with each other. So here on this diagram, you can see that they have this kind of counterclockwise twist. They're wrapping around each other, but the nitrogenous bases are on the inside of this helix. They're on the inside of the spiral. And they are lined up with each other in a very particular way. The nitrogenous bases actually hydrogen bond with each other. We call this base pairing. And each nitrogenous base is hyper-particular about which partners it's willing to pair up with. So in a DNA molecule, A is always found on one sugar-phosphate backbone across from T on the opposite sugar-phosphate backbone. C is always found across from G, C from G, T from A, C from G, T from A from T, curvy letters with curvy letters, angular letters with angular letters. A and T bind together, C and G bind together. Now these are hydrogen bonds, but in every single one of your cells you have over 6 feet of DNA, so there are a lot of hydrogen bonds holding these things together. When we want to get the information out of a DNA molecule, we start at one location and then we kind of unzip the entire double helix, breaking one set of hydrogen bonds at a time. Just like any super complicated modern video game could be broken down into binary code, ones and zeros and zeros and ones, all of the diversity of genetic traits that we see out in the population, all of that can be reduced down to A, T, C, G. It is the exact order in which these letters show up that codes for one version of a gene or another version of a gene. You have an AT here, that's a lactose tolerance gene. You have a CA here instead, that might be lactose intolerance. So the order is very, very important in DNA. Now, you already know that a dehydration reaction is used to combine two monomers to link them together in order to make polymers. So DNA molecules are made from nucleotides, and those nucleotides are linked through dehydration reactions. But for every kind of polymer, we have a different name for the bond that exists between the monomers. In the case of DNA, this is called a phosphodiester bond. I know you're thinking, I thought I was done memorizing different kinds of bonds. We had covalent bonds and ionic bonds and polar bonds and nonpolar bonds and hydrogen bonds. Why are you throwing more at me? It's just what this particular arrangement of atoms is described as. These are still covalent bonds, so they are sharing their electrons in these bonds, but having carbon, oxygen, phosphorus, oxygen, carbon like this is just referred to as a phosphodiester bond. I'm not going to ask you to draw a sugar-phosphate backbone with all of the elements in the correct place from memory or anything like that, but you are going to want to remember that the bond that exists between two nucleotides that are attached to the same sugar-phosphate backbone, that they're on the same strand, is called a phosphodiester bond. When we get to sugars, a bond between two monosaccharides is called a glycosidic bond, and when we get to proteins, a bond between two amino acids is referred to as a peptide bond. And speaking of proteins, there is a relationship between DNA and RNA and proteins, and here's what it is. A single chromosome is one long string of DNA, and on that string of DNA, you have a whole bunch of segments which we refer to as genes. You've probably heard of genes before. You've heard someone say, oh, that person's got good genes. They're so athletic and smart. Well, the genes are kind of what control your traits, but what they really do is code for different proteins, and then how those proteins work is what's going to control how you develop over the course of your life. So that's how they control your traits. The information is stored on the DNA molecule, but in order to actually make a protein out of it, we need to copy the DNA strand into an RNA message. So we copy it into that other molecule, RNA, just the section of the molecule that we want though, okay? So we'll only copy down one gene into RNA, and then that RNA will be used in order to code for a protein. And here you can see it's called a polypeptide. A polypeptide is a chain of amino acids, and then once that polypeptide is folded, you get a protein. So the next thing we're going to talk about is going to be proteins. You don't need to know transcription and translation yet. Again, we'll get to that later, but there is a relationship between these three types of molecules.